Hi everybody. Try to imagine your hand touching a hot pan. What reaction do you do? Surely, you will reflexively pull your hand quickly, blow or wet with water the part that is exposed to the heat. Have you ever thought about how this reflex mechanism occurs? How can the feeling of heat in the hands be sent to the brain so that we realize that our hands are exposed to the heat of the pan, and then we withdraw our hands to avoid being exposed to the heat longer? What happens if the heat information doesn't reach the brain? Surely our hands will blister badly, right? Well, of course, the information is sent by the axons of the sensory nerve cells in the hand to the brain. So, how do these axons send information? Each axon regenerates the transmitted impulse. Imagine the axons of our neurons are rows of people holding hands. The first person will squeeze the second person's hand. The second person will squeeze the third person's hand, and so on in sequence. The impulse will be transmitted along the row of people holding hands, and the strength of the impulse will not decrease because each person will renew the strength of the squeeze of the hand. This is an illustration of how the axon transmits impulse. Thus, the strength of the information will not be reduced to the destination. Although the axon's method of transmitting an impulse prevents a touch on your shoulder from feeling stronger than one on your toes, it introduces a different problem because axons transmit information at only moderate speeds, varying from less than 1 meter per second to about 100 meter per second. A touch on your shoulder reaches your brain sooner than will a touch on your toes, although you will not ordinarily notice the difference. Your brain is not set up to register small differences in the time of arrival of touch messages. After all, why should it be? You almost never need to know whether a touch on one part of your body occurred slightly before or after a touch somewhere else. Okay, let's talk about it. After knowing the basic components of nerves, namely neurons and glia, we will study how the nerve cells transmit information to each other so that we can experience mental processes. In this discussion, we will examine the mechanism of transmission of information by nerve impulse, namely from resting potential, axon potential propagation, myelin sheath and saltatory conduction, and knowing what a local neuron is. Information can be received and transmitted by neurons due to changes in resting potential. So let's first learn what resting potential is. You need to know the entire body of the neuron is covered by a membrane as thick as 8 nanometers. That is about 1/10,000th the width of a human hair. This membrane consists of two layers of phospholipid molecules consisting of a weak acid chain and a phosphate group. The phospholipids are also attached to cylindrical protein molecules. These cylindrical proteins allow chemical to pass through the membrane. At rest, the membrane regulates what is called the electrical gradient or polarization, namely the difference in electric charge between the outer and inner regions of the cell. The electric potential inside the membrane is usually negative when compared to the outside of the cell. This is due to negative proteins contained in the cell. The difference in voltage between the outside and inside of the cell is called the resting potential. How can researchers measure this resting potential? By inserting very thin electrodes into the cell body. The electrode is connected to a voltmeter, so we can know the potential inside the neuron is negative. The magnitude is approximately minus 70 millivolts. At resting potential, the charge inside the neuron is negative when compared to the outside of the neuron, because inside the membrane there are more negatively charged proteins. This happens because of the sodium-potassium pump mechanism. What is that? So the neuron membrane is selective permeable, meaning that there are chemical elements that can pass freely but some cannot. Substances that can pass freely include oxygen, carbon dioxide, urea, and water, while other important ions such as sodium, potassium, calcium, chloride must pass through the channel, a membrane gate, to be able to enter and exit the neuron. You can see in this picture there is a channel gate on the membrane. When the membrane is at rest, the sodium and potassium channels are closed so that only a small amount of potassium can be passed and no sodium can pass at all. With stimulation, these channels can open and let sodium and potassium in. This stimulation causes what is called the sodium-potassium pump. As the name suggests, the sodium-potassium pump is tasked with pumping sodium and potassium in and out of the membrane, but in different directions. Let's look at this picture. Here's a sodium-potassium pump. Either is a transmembrane or a protein complex. Let's remember, Na is sodium and K is potassium. So the sodium-potassium pump is an active transport that requires energy to carry out its duties. To carry out this action requires adenosine triphosphate as energy to change the concentration gradient of ions. Well, sodium-potassium pump usually occurs in cells, where the environment in the cell requires more potassium than sodium. So retain in the cells the amount of potassium more, while outside the cell, the amount of potassium is maintained less than inside the cell. So to maintain this, sodium in the cell needs to be removed, while potassium needs to be added, so that the amount of sodium and potassium is balanced, both outside and inside the cell. This is outside and this is inside the cell. The steps for the sodium-potassium pumps are as follows. First, open channel on the inside of the cell. Sodium enters the attachment channel as much as 3 molecules. Hydrolysis of adenosine triphosphate into adenosine diphosphate and phosphate attached to integral proteins. When phosphate attached, the process of phosphorylation occurs. The channel will open out. Sodium will leave the cell as much as 3 molecules. Potassium enters the channel, binds the protein as much as 2 molecules. Phosphate is released and protein opens inward. Potassium is released into cells. After the potassium is released, it is repeated again to number 1 to maintain more K+ condition in the cell. The result of the sodium-potassium pump is that there are 10 times more sodium ions outside the membrane than inside the membrane. The job of the sodium-potassium pump is to restore the resting potential. The body invests much energy to operate the sodium-potassium pump, which maintains the resting potential. Why is it worth so much energy? The resting potential prepares the neuron to respond rapidly. As we shall see in the next session, excitation of the neuron opens channels that allow sodium to enter the cell rapidly. Because the membrane did its work in advance by maintaining the concentration gradient for sodium, the cell is prepared to respond vigorously to stimulus. Compare the resting potential of a neuron to a poised bow and arrow. An archer who pulls the bow in advance is ready to fire at the appropriate moment, right? The neuron uses the same strategy. The resting potential remains stable until the neuron is stimulated. Ordinarily, stimulation of the neuron takes place at synapse, which we consider in the next topic of this course. In the laboratory, it is also possible to stimulate a neuron by inserting an electrode into it and applying current. As we know that communication between neurons occurs through the axons under resting potential, they have a magnitude between less than 1 millivolt to 5 millivolt per second. This is of course normal in the short communication between neurons, but now imagine if it were the neurons in the feet. When our feet get sensation, it takes some time to convey information to the brain. If the magnitude is short, it can be imagined that the information will be cut off in the legs and not yet reach the brain. So to speed up sensation information from the tip of the big two, we need a higher magnitude resulting from the action potential that can reach 100 millivolt per second, so that information from the big two can immediately reach the brain. In this case, to make the faster process in delivering information to the brain, there is an insulator that we've already learned in the previous session. This insulator called as myelin. Can you remember? The axon covered by myelins. Messages sent by axons are called axon potentials. When the membrane is completely depolarized, reaching the threshold, the sodium and potassium channels open. Sodium ions enter the cell rapidly and reduce and reverse the change in the membrane. This event is called as an action potential. Please have a look the illustration of an action potential. To understand action potential, let's begin by considering what happens when the resting potential is disturbed. We can measure a neuron's potential with a microelectrode. When an axon's membrane is at rest, the recording shows a negative potential inside the axon. If we now use a different electrode to apply a negative charge, we can further increase the negative charge inside the neuron. The change is called hyperpolarization, which means increased polarization. When the stimulation ends, the charge returns to its original resting level. The recording looks like this. Now let's apply a current to depolarize the neuron, that is, reduce its polarization towards zero. If we apply a small depolarization current, we get a result like this. With a slightly stronger depolarizing current, the potential rises slightly higher, but again returns to the resting level as soon as the stimulation ceases, like this. Now let's apply a still stronger current. Stimulation beyond the threshold of excitation produces a massive depolarization of the membrane. When the potential reaches the threshold, the membrane opens. Its sodium channels and let sodium ions flow into the cell. The potential shoots up for far beyond the strength that the stimulus provided, like this. Any subthreshold stimulation produces a small response that quickly decays. Any stimulation beyond the threshold, regardless of how far beyond, produces a big response like the one shown, known as the action potential. The peak of the action potential shows as 130 millivolt in this illustration, varies from one action to another. Note that any depolarization that reaches or passes the threshold produces an action potential. For a given neuron, all action potential are approximately equal in amplitude or intensity and velocity. That is, the intensity of the stimulus cannot cause a neuron to produce a bigger or smaller action potential or a faster or slower one. Slight variations can occur at random, but not because of the stimulus. More properly stated, the all or none law is that the amplitude or velocity of an action potential are independent of the intensity of the stimulus that initiated it, provided that the stimulus reach the threshold. The strength of the stimulus does not affect the velocity and intensity of the action potential. As long as the potential reaches the threshold point, the action potential will occur at the same velocity and intensity it called as all. If it does not reach the threshold, the neuron will not be depolarized. It calls none. By analogy, imagine flushing a toilet. You have to make a press of at least a certain strength, the threshold. But pressing harder does not make the toilet flush faster or more vigorously, right? Similarly, when you flick the switch to turn on the lights in your room, flicking the switch harder will not make the lights brighter. The chemical events behind the action potential may seem complex, but they make sense if you remember three principles. At the start, sodium ions are mostly outside the neuron, and potassium ions are mostly inside. When the membrane is depolarized, sodium and potassium channels in the membrane open. At the peak of the action potential, the sodium channels close. The term propagations of the action potential describes the transmission of an action potential down an axon. Action potential occurs successively along the axon. As sodium ions flow through the center of the axons and stimulate the next point along the axon toward its threshold. This propagation keeps the action potential the same size along the neuron. As an action potential occurs at one point on the axon, enough sodium enters to depolarize the next point to its threshold, producing an action potential. At that point, in this manner, the action potential flows along the axon, remaining at equal strength throughout, behind each area of sodium entry potassium ion exits, restoring the resting potential. Immediately after the action potential, the membrane enters a refractory period. During this period, the membrane refuses to initiate another action potential. Remember, at the peak of the action potential, the sodium gets snapshot. As a result, the cell is in a refractory period during which it resists the production of further action potential. In the first part of this period, the absolute refraction period, the membrane cannot produce another action potential regardless of the stimulation. During the second part, the relative refractory period, a stronger than usual stimulus is necessary to initiate an action potential. The refractory period depends on two facts. The sodium channels are closed and potassium is flowing out of the cell at a faster than usual rate. In myelinated axons, action potential occur only at the nodes. Myelin covers the axons, so the electrical code doesn't leak. Jumping of action potential from node to node is referred as saltatory conduction. Speed up impulse transmission and less energy. Axon produce action potentials. However, many small neurons have no axon. Neurons without an axon exchange information with only their closest neighbors. We therefore call them local neurons. Because they do not have an axon, they do not follow the all or none law. When a local neuron receives information from other neurons, it has a graded potential, a membrane potential that varies in magnitude in proportion to the intensity of the stimulus. The change in membrane potential is conducted to adjacent areas of the cell in all directions, gradually decaying as it travels. Those various areas of the cell contact other neurons, which they excite or inhibit. Local neurons are difficult to study because it is almost impossible to insert an electrode into a tiny cell without damaging it. Most of our knowledge, therefore, has come from large neurons, and that bias in our research methodology may have led to a misconception. Nonsensical belief that they say we use only 10% of our brain. This belief has been remarkably persistent given its total lack of justification. What does it mean? Does it mean that you could lose 90% of your brain and still behave normally? Of course not. If you could contract all your muscle at one time, you would not be a great athlete. You would just have spasms. If you could active all your neurons at one time, you would not have brilliant thoughts. You would have an epileptic seizure. Any meaningful thought or activity requires activating some neurons and inhibiting others. And the inhibition is just as important as the excitation. You use all of your brain, regardless of whether you are using it well. Thank you for watching, and now it's time for you to go to Ella and complete the assignments there. Have a great time.